Ever wished we could fix diseases the way we fix typos? That's exactly what CRISPR-Cas9 promises, a tool that lets scientists edit DNA with remarkable precision. In this video, we'll break down what CRISPR is, how it works, and why it's being hailed as a potential game-changer in the fight against cancer. Over the past decade, CRISPR has been making waves for its ability to rewrite genetic code. At its simplest, think of it as a programmable molecular scissor able to cut, remove, or replace pieces of DNA exactly where scientists want. It all starts with the guide RNA, which acts like a GPS, leading the Cas9 enzyme to a specific spot in DNA. Cas9 then makes the cut, and the cell's repair systems take over. Sometimes the repair is messy, which is useful for switching off harmful genes. Other times, scientists give the cell a template so it can fix or replace the sequence with precision. This isn't just a theory. CRISPR is already being studied to correct genetic disorders, supercharge immune cells, and even target the mutations that drive cancer. And today, we're going to see how it all came together, the challenges researchers face, and where the future might take us.
Before delving into the fascinating world of CRISPR, let us first uncover the story of how it all began. In 1987, Ishino and colleagues discovered unusual DNA repeats in E. coli, the first glimpse of what we now call CRISPR. By 1995, Francisco Mojica found similar repeats in archaea, suggesting a conserved biological role. In 2002, Janssen's team officially coined the term CRISPR and identified nearby Cas genes. By 2005, several researchers proposed that CRISPR acted as an adaptive immune system after realizing its spacers matched viral DNA. This theory was confirmed in 2007 when Barango and Horvath proved that Streptococcus thermophilus could acquire viral resistance through CRISPR. A major leap came in 2011 when Charpentier's group discovered tracer RNA, a crucial component for Cas9 activation and RNA-guided DNA cutting. In 2012, Charpentier and Doudna engineered a single guide RNA by fusing cRNA and tracer RNA, enabling programmable gene editing with Cas9. Then, in 2013, the breakthrough year, CRISPR-Cas9 was successfully used to edit DNA in human, mouse, and zebrafish cells. Techniques were refined with codon-optimized Cas9, nuclear localization signals, and homology-directed repair, making CRISPR efficient and reliable. This marked the transition from theory to practical application, paving the way for diseases modeling, gene therapy, and agriculture innovation. Then, in 2014, the first crystal structures of Cas9 were published, revealing its precise interaction with guide RNA and target DNA. And lastly, in 2020, Emmanuel Charpentier and Jennifer Doudna received the Nobel Prize in Chemistry, recognizing CRISPR-Cas9 as a game-changing genetic tool.
Like what was previously discussed, CRISPR-Cas9 offers various beneficial applications aimed at combating both hunger and disease, including certain types of cancer. Rabaan et al. 2023 identified a limited number of cancer types and their associated CRISPR mechanisms, including liver cancer, colorectal cancer, renal cell carcinoma, bladder cancer, and pancreatic cancer. These cancers primarily utilize CRISPR-Cas9 for tumor modeling, functional genomics, and immune modulation. In the case of liver cancer, the initial target gene is FAH, or fumarate acetoacetate hydrolase gene. It is essential for tyrosine metabolism in the liver. Mutations in this gene lead to a rare metabolic disorder called hereditary tyrosinemia type 1, which if left untreated progresses to liver failure. A geneticist will first target the FAH gene, which is responsible for breaking down tyrosine in the liver. They will design and utilize sgRNA specific to the FAH gene to guide the CRISPR-Cas9 system to the exact mutation site. The sgRNA will be combined with a donor DNA template carrying the correct FAH sequence to repair the mutation. This preparation step ensures that both the editing tool and the repair template are ready for delivery. Using hydrodynamic tail vein injection, the CRISPR-Cas9 complex together with the donor DNA will be delivered directly into the liver cells. Once inside, Cas9 will create a double-stranded break at the mutant FAH site. The cell's natural repair system will then use the donor DNA to correct the mutation, restoring the normal FAH gene. With the gene restored, liver cells regain the ability to metabolize tyrosine normally, preventing toxic buildup and restoring healthy function, effectively removing the underlying cause of liver damage and reducing cancer risk. However, this same experiment also revealed a significant challenge. While attempting to correct the mutated FAH gene, the geneticists simultaneously targeted the P10 and P53 genes. Cas9 induced double-stranded breaks in these genes, and through non-homologous end joining, both were knocked out. The loss of P10 and P53 led to tumor formation, providing insight into how liver tumors develop and progress. This highlighted that aside from correcting mutations, the unintended loss of tumor suppressor genes can trigger cancerous growth.
Up next, the challenges of CRISPR. Despite its promise, CRISPR still faces challenges that limit its application in medicine. One major concern is off-target effects, where unintended DNA regions are edited, potentially causing harmful mutations. Another issue is immune response. Since CRISPR uses bacteria-derived Cas enzymes, the body may recognize them as foreign, triggering inflammation or destroying edited cells. This also raises the risk of immune rejection in repeated treatments. Because of this risk, CRISPR is currently used only in select treatments, and further research is needed to expand its safe and effective use.
While CRISPR-Cas9 has already shown promise in cancer research, scientists believe the next step is to make it safer, more precise, and more widely applicable. First, researchers aim to expand CAR-T therapy beyond blood cancers, engineering immune cells to attack solid tumors and release immune-boosting signals right where they're needed. Second, large-scale CRISPR screens will help pinpoint the genes cancer cells get active without, revealing new targets for treatment. Next, RNA targeting tools like Cas13a could allow for reversible edits, silencing dangerous mutations without permanently altering the DNA. Fourth, controllable CRISPR systems activated by light or ultrasound would give doctors precise on-and-off control, reducing risk and side effects. And finally, integrating AI into CRISPR design could help speed up discoveries, create highly accurate gene guides, and prevent unwanted edits. By combining these innovations, CRISPR could evolve into one of the most precise and reliable cancer treatments of the future. Looking ahead, the ultimate goal of CRISPR in cancer therapy is to achieve highly precise, low-risk, and personalized treatments. Researchers envision a future where a patient's unique genetic profile guides custom-designed CRISPR edits potentially used alongside chemotherapy, radiation, or immunotherapy for stronger multi-pronged cancer eradication. Though the technology is still young, its potential is vast, offering not only treatment, but the possibility of preventing or even eliminating certain cancers entirely. CRISPR-Cas9 has transformed our approach to science, medicine, and disease treatment in less than a decade. While challenges remain, the promise it holds is immense. With continued research, ethical use, and innovation, the dream of editing cancer out of existence could become a reality in our lifetime.